Hello everybody, in this video we're going to be looking at body plan development and all of the genes that are involved in this and the effects that they have. Now I know this is only a small part of the specification, but it has come up on the exams before, so we do need to pay careful attention to this. To make sure you've got all of the genes and the specific bits really sorted in your head, over the website Dr. Upwards has given you a special set of questions just to go with this video.
Body plans and development. The homeotic genes are a large family of genes that control anatomical development. This is really important so that all structures develop in the correct location according to the body plan. So if you think about this human I have here, so we've obviously got our head, our eyes, all the features on the face need to be there on the face, and you've got your muscles need to be in the right place, all the things in your abdomen need to be in the right place, or your organs, all the way down to kind of your fingers at the end of your hands, your toes at the end of your fingers. All of that is the body plan, like where all of these structures sit in relation to each other along kind of the body from head to toe. Some of these homeotic genes contain Homeobox sequences. So these are about 180 base pairs of DNA long, and they code for a 60 amino acid sequence. They produce proteins, so the 60 amino acid sequence is a protein, and these are proteins that bind to DNA at the promoter region and act as transcription factors. So we've talked about transcription factors in another video. Make sure you watch that first before we kind of think about how this works. But we've got that promoter region, and we've got that what we call the tata box, which is that kind of region of bases where proteins such as transcription factors know where to bind in order to then trigger this transcription of these genes. So these proteins that come from the Homeobox sequences are specifically transcription factors, which will then control the expression or the regulation of other genes, which is what controls this physiological development according to the body plan. So Homeobox genes produce homeodomain proteins, and these regulate gene expression relating to the development of structures in the body. These Homeobox sequences are highly conserved. What we mean by highly conserved is that they have remained similar, so the actual base sequences remain similar in all descendant species throughout evolution. There's molecular evidence that we've now seen that has shown that these genes are present all the way back to jellyfish, or a family known as the cnidarians. So if you look at my evolutionary tree here, that's way back in time. That's before insects, that's before mollusks, snakes, crabs, way before mammals, and that's about 541 million years ago. These sequences would have been present in organisms that are not symmetrical in terms of what we think about bilateral symmetry. Jellyfish are very simple organisms. You wouldn't think they'd have much of a sort of body plan. But it shows that if the sequences have remained pretty similar all the way as far back as that, it shows how important these genes are for regulating and developing organisms in the right way, from embryos or kind of small organisms up until like their fully grown size, to make sure that all of their structures and tissues are going to differentiate and organize themselves in the right way. There is a specific group of Homeobox genes called the Hox genes, and these are found only in animals. They regulate the development of body parts from head to tail along what we call the anterior to posterior axis. If these Hox genes are mutated, it can lead to body parts developing in the wrong places. This can be seen in flies when they can sometimes have things like legs on their heads instead of their antennae. Hox genes are themselves regulated by genes called gap genes and parallel genes. And these are controlled by mRNA that's found in the egg cytoplasm. So it's something that's passed down from maternal parents that contains this mRNA that's found in the egg cytoplasm that is going to contain these or code for these proteins that are going to control the gap genes and the parallel genes, and then those expression of the gap genes and the parallel genes will control the expression of the Hox genes. So it's kind of this whole cascade of kind of control of gene expression which regulates how these genes work. The Hox genes are activated in developing embryos, and they're expressed in order along the anterior-posterior axis. So they are actually expressed not only just in a sequence, so what comes first, but also in time order. So as each one is expressed in sequence and in time, that's controlling what in time is developing first in terms of the physical body of the embryo. So it controls the order of the development of parts of the body. So we've got the head, the anterior thorax, the posterior thorax, and the abdomen. That's in the adult. You can see at the top there. And then we have the embryo, and you can see I've labeled kind of the 1, 3, 5, 8. That tells you that that part of that gene, which is red, that will be being expressed. That will be expressed first. And then that part of the body, the kind of face, head part, will be activated. Then 3, the yellow kind of the part of the head with the eyes and everything, will then come next. Then 5, the thorax, the anterior thorax. Then 8, the abdomen. And it's been directly related. Those sections that are coloured have been directly related to those genes that are coloured. And we know that they specifically code for that region of the body development. In a similar way to what we talked about with the other Homeobox genes, Hox genes code for homeodomain proteins, and these act as transcription factors. What these transcription factors actually control is mitosis, apoptosis, cell migration, and the cell cycle regulation. So the control of mitosis in the cell cycle. This makes sense because during development, obviously, mitosis is going to have to happen because as an embryo grows, it's going to need more cells. And the control of kind of how many cells are growing, how fast they're growing, if cells need to go through apoptosis or if cells need to move somewhere, all of that control by these genes is how we're going to develop an embryo from a small embryo into the fully sort of grown organism with all of the tissues and differentiated cells that it needs. So normally body cells divide around 50 times before dying. So the regulation of this cell division is really important. And the Homeobox genes play a role in this regulation of how often or how long a cell is dividing before its time is up. And then that cell would normally go through apoptosis. There are other reasons why a cell might go through apoptosis, otherwise earlier or for some other reason to help with the development of this body plan. And that's what we're going to look at next. Apoptosis and mitosis in development. Programmed cell death or apoptosis is the normal, controlled breakdown of cells during development. The first stage is where enzymes begin to break down the cell contents. The chromatin condenses and the cell generally shrinks. Then the cell membrane starts to change and protrusions form called blebs. The cell begins to fragment and break into vesicles. Phagocytes then engulf and digest the cell fragments. Mitosis and cell differentiation create the majority of the body plan. But mitosis is needed to refine that body plan. It helps by removing certain tissues or unwanted cells. For example, in human development, it separates fingers and toes. If this is not done, then they can remain fused or webbed. It is also the way metamorphosis happens in things like tadpoles that turn into frogs. The tail is removed. It's not lost, it's broken down through apoptosis. Genes that regulate mitosis and apoptosis are only expressed in appropriate cells during development, so they're switched on and off depending on what the cells need to do at that point. Their expression can be influenced by both internal and external stimuli. For example, if a cell's DNA is damaged and apoptosis does not occur, then tumours may form.
Ouch! This is why in some videos I explain scratches.